question 6. Okay, question 6. The sodium-potassium pump is a membrane protein found in animal cells. Give one function of sodium-potassium pumps. Okay, they are super, super important. It's all about changing ion gradients. So you've got ion concentrations are different inside and outside. So you've got maintaining an ion gradient. So if you've got an ion gradient, oh, sorry, wrong one in. So once you've got your ion gradient, you then use that for different things. Okay, so so once you've produced that, and you had to get all of this in, so once you produce that, you can use that either for in your kidneys, kidney tubules. You can use that for glucose transport, but more specifically, you're going to have to say symport, because that's moving with the ions. And also for nerve cells. Oh, nerve cell potential. Okay, you needed to give, as I say, this bit and one of these with it, or you could say it's about osmotic balance. So maintaining that within the cell. I'm afraid you just had to be giving that quite precisely from your content set. Okay. Describe the role of ATP in altering the affinity of the pump for sodium ions. Okay. When in doubt, anything to do with proteins, you're like, it's a conformational change. And that would get you the mark, basically. Okay, so ATP phosphorylates the pump, which gives conformational change. Conformational change. Okay, now once you've got a conformational change, then that means that the sodium ions affinity drops, lowers, changes. Okay, and vice versa. When your pump dephosphorylates, and you lose that phosphorylation, then the conformational change that flips it back means that the sodium ion affinity increases again. Okay. Digoxin is a chemical that inhibits the sodium-potassium pump by binding to the potassium ion binding site as shown in the diagram. Explain why binding by digoxin prevents further binding of sodium ions by the pump. Okay. Well, if you've got digoxin in here, okay, and this point was actually the potassium site, the potassium can't bind here. If the potassium can't bind, then potassium action doesn't happen. So if the potassium doesn't bind, then the potassium doesn't move through, which means you don't get dephosphorylation, or you don't have your conformational change that you need. So sodium-potassium affinities just remain the way they were. And at the moment, it's sitting there with a low affinity for sodium and a high affinity for potassium, but that high affinity for potassium has been completely sidetracked by the digoxin going in. Okay, so 2 marks. You can't get the potassium to bind, and that means that you cannot get the conformational change by dephosphorylation. And that's that question.